Microbes and dead cells could trigger inflammatory mediators to cause inflammation, which is basically the body's response of delivering leukocytes and plasma proteins to the site of infection or injury so that the offending agents could be removed or eliminated. When the body is inflamed due to microbes or other external agents, leukocytes migrate from the blood vessels to the site of infection through a process called chemotaxis. Once in the site of infection, leukocytes have to recognize the offending agents in order to be activated and to eliminate them. Once activated, the leukocytes perform various functions to eliminate the offending agents or microbes. Leukocytes possess numerous receptors for recognizing external agents, some of them being toll-like receptors and G-protein coupled receptors. They also possess receptors for other proteins like cytokines, which could activate them to start the cleansing process. Once activated, leukocytes perform various functions to amplify the process of inflammation. That is, it could cause further migration of leukocytes to the site of infection and help in production of various inflammatory mediators like cytokines and arachidonic acid metabolites. Now, apart from amplifying the inflammatory response, leukocytes also eliminate offending agents like microbes by ingesting and destroying them, a process called phagocytosis. Phagocytosis involves 3 steps, them being the attachment of the offending agent or microbe to the phagocyte receptors, engulfment of the offending agent or microbe, and intracellular killing of the offending agent or the microbe. There are receptors like mannose and scavenger receptors on phagocyte, which can bind to microbes and help in engulfment. Another important receptor is the opsonin receptor, which helps in binding to opsonins that have coated the cell surface of microbes. For example, immunoglobulins like IgG and complement proteins like C3b act as opsonins that could bind to cell surface of microbes, which in turn can be recognized by opsonin receptors on phagocytes to help in engulfment of the microbe. After attachment to phagocyte receptors, the plasma membrane of the phagocyte through extensions called pseudopods around the microbe engulfing it. Now this part of the plasma membrane gets pinched off, forming a vesicle called phagosome with the microbe inside. The phagosome then fuses with a lysosome inside the cell, forming phagolysosome. The killing or elimination of microbes is accomplished by chemicals called free radicals, which are essentially of 2 types, the reactive oxygen species and the reactive nitrogen species. Reactive oxygen species are derived by reduction of oxygen by an enzyme called phagocyte oxidase or NADPH oxidase. And reactive nitrogen species are derived from nitric oxide combining with superoxide. Phagocyte oxidase present in the cytoplasm translocates to the membrane of the phagolysosome and reduces oxygen to a superoxide anion. The superoxide anion is converted to hydrogen peroxide, which is further converted to hypochlorite by myeloperoxidase. Hydrogen peroxide is also converted to a hydroxyl radical. Now, hypochlorite and hydroxyl radical are powerful destructive agents helping in elimination of microbes. Further, nitric oxide derived from arginine by the action of nitric oxide synthase combines with superoxide to form a free radical called peroxynitrite, which also participates in microbial killing. The free radicals target the lipids, proteins, and nucleic acid of microbes, thus killing them. Now, apart from free radicals, phagocytes also possess numerous enzymes like lysozyme, elastase, cathelicidin, lactoferrin, and defensins, which are antimicrobial.